to our first speaker from the external users of the both EBIC and B24. So the first person is going to be Vicki Gold from the University of Exeter. So Vicki originally did a PhD in Bristol, where she was working on secretory pathways in bacteria, and then she went on a long-term fellowship to the Max Planck in Frankfurt. So it's a very, very good place. And she's now back in the UK in Exeter with her own lab doing lots and lots of interesting things, which I'm sure she's going to tell you about.
Okay, thanks, Dan. I'll just tell me if this is all good to go.
Yep, looks good.
Very good. Okay.
Yep.
So firstly, thank you very much for the invite to present the data today. I'm going to tell you about type 4 pili in bacteria. And the work started out when I was a postdoc in the Coolbrand lab at the Max Planck Institute of Biophysics in Frankfurt, and it has been continued in my lab in Exeter, where I've been a PI for the last 3 years. The title is A New Twist on Type 4 pili, Cryo-EM reveals 2 distinct filaments assembled by the same bacterium. So firstly, what are type 4 pili? Well, they're long filamentous appendages which project from the surface of the cell, and they're used for motility. So they work in cycles of extension, adhesion of a tip to a surface, and retraction from the base, which leads the cell to move forward in a jerky form of motion, which is referred to as twitching, which is what you can see in this video. Both extension and retraction are powered by ATPases. So we're interested in the machinery that assembles type 4 pili and how this works. And this is a schematic of the machinery in Thermus, which is our model system. So structures are known for some parts of this machinery, and I'll go through some of them, but I haven't covered everything. So firstly, we have PIL-Q, and this is a secretin protein. It forms a channel in the outer membrane through which the type 4 pilus can pass. And the platform at the inner membrane is comprised of PIL-C, PIL-M, PIL-N, and PIL-O, and these are required for assembly of the filament. There's structure known, for example, of soluble parts of some of these proteins, so the cytoplasmic part of PIL-M with a PIL-N peptide, and the periplasmic part of PIL-N. And we also have the ATPases. So as I mentioned, these are required for assembly and disassembly of the filament. And PIL-F is used for assembly in Thermus. And we have PIL-T, which is required for disassembly. In Thermus, there are 2 PIL-T enzymes, PIL-T1 and PIL-T2. So the type 4 pilus machinery also plays a role in DNA uptake, and this is by interacting with these common proteins, which are shown here at the cytoplasmic membrane. And this is a very interesting mechanism for us because this mechanism of DNA uptake obviously enables genetic adaptation through horizontal gene transfer and can allow the development of pathogenicity and antimicrobial resistance. So, however, the mechanism of action, so how this machinery can perform 2 seemingly independent functions, is not clear. So I'm going to talk briefly about my first project in this system, which was to study the entire type 4 pilus machinery in situ by electron tomography. And this is the work that I carried out as a postdoc. Now, I wasn't going to give any background to the methods for this audience because I didn't think it would be necessary, but then when I swapped sessions and became one of the first to speak, I thought I'd better put in a little bit of methods, at least for those who are new to cryo-EM in the audience. So it's very brief. So our cells are grown and then are pipetted onto EM grids. We then plunge freeze them in liquid ethane to vitrify the sample and transfer them into the electron microscope. Now in tomography, we tilt the sample in the microscope and collect an image at every tilt. And then these 60 or so images are then processed to a 3-dimensional volume. We then select our proteins of interest, which can be extracted, aligned together, and a structure can be determined by subtomogram averaging. So here is a tomogram of a Thermus thermophilus cell. And you can see in the segmented version, we have numerous pili extending from the cell surface, and they're concentrated primarily at the cell poles. On close inspection of the membrane region, we can identify protein complexes in the periplasm, of which we could see two forms. On the left, we have complexes in the absence of a pilus, and on the right, in the presence of a pilus. So as I mentioned in the previous slide, we could extract these subvolumes, align them and average them together to generate two structures. And this is the result. So we have a closed state complex and an open state complex in the presence of the filament. And I'll just talk you through a little bit about these. So we can see PIL-Q, which is the secretin embedded in the outer membrane, and it has two gates which are closed in the closed state. And you can see that PIL-Q interacts with a number of other proteins, which we've just called C1, P1, and P2, because C for cytoplasmic and P for near the peptide glycan. We don't yet know the precise identity of the proteins which constitute these densities. You can see there's quite some conformational changes which have occurred on going into the open state. Of course, the gates have opened, the pilus is assembled through the center, and the N-terminal parts of PIL-Q, which you see here, have shifted down towards the cytoplasm. Of interest, we also identify some additional density present in the open state, which was not in the closed state, and we speculate that these may be the ATPases, which are required to drive filament assembly and disassembly. So here is a segmented, or rather a 3D version of these structures, and a very low resolution structure of the filament just docked into the center. This was determined by subtomogram averaging, and there'll be a bit more on that later. I can now just show you a morph movie going between these two different conformational states, so you can appreciate the large scale changes which must occur in this whole system. So as an aside to this work, we made an interesting observation, and that's that sometimes the filaments which emerge from the type 4 pilus machinery look different. So on the left, we have filaments which looked wider than those which I show on the right, indicated in orange, the wide ones indicated with blue arrowheads. Now, there are no other secretins or pilus assembly proteins encoded on the Thermus genome. So we thought it was likely that they are both type 4 pili. It's very hard to see differences by cryo-electron tomography. It's not super high resolution. So in the next part of the talk, I'm going to talk to you about high resolution structures of the filament, which are based on single particle electron microscopy and helical reconstruction. So what do we know about type 4 pili? Well, they are comprised of thousands of copies of individual protein monomers, which are called pilins, and they are assembled into a helical filament. There are 5 structures already known in the 5 to 12 angstrom resolution range. So our aim of this work was firstly to see if we do indeed have 2 different filaments, and secondly to determine their structures at a resolution which would be sufficient for atomic model building. So the sample preparation is pretty straightforward. Cells are grown on plates and then they're scraped off. And then the purification of pili is fairly crude, but it works. So they're removed by ammonium sulfate precipitation, centrifugation, and then you can resuspend the filaments in the buffer of your choice. The grids are then again vitrified by plunging into liquid ethane prior to electron microscopy imaging. So we optimized our grids in Exeter, which was the bulk of the work. We then screened them at the GW4 facility for cryo-EM in Bristol, and then we collected our final data set at EBIC on Titan Cryos. So I'm going to just give you a really brief background to the methods that we used. So we collected our data in movie frames and we aligned those, and then selected the filaments, which are then boxed into sections. This is then followed by 2D classification to remove the poorly aligned particles. The power spectra is then used to determine the helical parameters, which can be fed into 3D classification. The most promising class then goes on to 3D refinement, from which we can then calculate our resolution and go on to atomic model building. And these are some examples of the images which we obtained at EBIC. So now you can quite clearly see that there are both wide and narrow pili in these images. And these are the resulting 2D classes underneath. So that's really easy to discriminate. And conveniently, of course, we have two structures then from one data set. And these are the resulting maps. So both filaments follow the conserved type 4 Pilus blueprint, and that's that they have a conserved bundle of alpha helices in the center, which are hydrophobic. And these are surrounded by a beta strand rich domain, which is exposed to the solvents. The wider Pilus measured about 70 angstroms across, and the narrow filament approximately 45 angstroms. And the two filaments had different helical rises and twists. So of course, we're interested to know the protein composition of the two filaments. And what we can do, something quite simple, firstly, is to extract an individual pilin monomer from the entire filament. And you can already see that it looks like these are going to be different proteins. The narrow pilin is much longer, and this is mostly due to this unfolded stretch, which you can see indicated in brackets. It's much longer for the narrow pilus than for the wide pilus. And our collaborators had shown in the past that there were 4 pre-pilin-like proteins previously identified in Thermus in one locus. These were called pili 1, 2, 3, and 4. Pili 4 was designated as the major pilin because when it was knocked out, cells didn't express pili anymore. And pili 1, 2, and 3 were designated as minor pilins with an unknown role in assembly or function and not necessarily a structural role. So in parallel, we had performed some mass spectrometry on our isolated Pilus preparations. Again, work from our collaborators had shown that cells are hyperpileated at low temperature. For Thermus, that means less than 60 degrees C. The optimal growth temperature is 68 degrees C. So that's why we perform these experiments at 2 different temperatures, 58 degrees C in blue and 68 degrees C in red. So the details of this graph are not important. The most important point to note is that by far the most abundant proteins in our preparations are pili 4 and a previously uncharacterized protein called TTC-1836. When we look at the sequence of TTC-1836, it's very similar in sequence to pili 4, and we didn't detect the minor pilins pili 1, 2, and 3. Now, the resolution of our EM maps was sufficient to build atomic models, and guided by these mass spectrometry results, we could fit the sequence for pili 4 into the wide pilus and the sequence for TTC-1836 into the narrow pilus. We now propose that TTC-1836 be called pili 5. You can see this unfolded stretch again more clearly in the atomic models, and a couple of other structural features which I'll point out. So in the beta strands, for pili 4, there are 4 strands, whereas for pili 5, there are 5. And they also have different interaction partners in the filament. So pili 4 interacts with 6 other pilins in the filament, whereas pili 5 interacts with 7. Most of these interactions are hydrophobic, which makes quite a lot of sense because they're non-specific and allow for sliding, stretching, and bending in the function of these filaments. And both filaments' disulfide bonds stabilize the C terminus, and there are a number of inter- and intramolecular salt bridges. So an interesting question, of course, arises as to what's the function of these two filaments. And we performed some imaging of 3 different samples. So we have our wild type, which we know has wide and narrow pili, and we could perform genetic disruption of the genes for pili 5, comprising the narrow filament, and pili 4, comprising the wide filament. And these images are from whole cells which have been negatively stained, and we could then perform 2D classification of the filaments. And we found this to be quite an accurate way of looking at the proportions of wide and narrow pili. And in fact, it's quite hard to see by eye, which could also be why this hadn't been noticed before. So in the 2D classes for the wild type, you could see that we had both wide and narrow pili as expected. When we disrupt PIL-A5, we had only wide pili, and that makes sense because PIL-A5 comprises the narrow filament. And when we disrupt PIL-A4, we don't have any pili whatsoever. Now, this wasn't surprising because this had been shown in the past that PIL-A4 mutations, cells do not express pili anymore. But being as PIL-A4 only constitutes the wide filament and not the narrow one, it tells us that this protein must also have a role in assembly or priming for both types of filament. So next, we performed some functional assays, the first being motility. And you could see that only the wild type cell is capable of movement. So this tells us that if neither of our mutants have narrow pili, it tells us that the narrow ones are required to promote cell movement. We could also perform some natural transformation assays looking at DNA uptake, again with the same samples and a control without DNA. And you can see that the mutant which has only wide pili was comparable to the wild type with both. So that tells us that the wide pili are sufficient for DNA uptake and that the narrow ones are not required. So I'd now like to show you a little bit about post-translational modification of our structures. So in both filaments, we could observe unaccounted for density in the EM maps, which couldn't be attributed to, say, an amino acid side chain. And these were found on serine residues, which is a pretty strong indicator for O-linked glycosylation. Type 4 pili have been shown in the past to be glycosylated, so this isn't necessarily a surprise. But interestingly, we find 3 on PIL-A4 and only 1 in PIL-A5. It was a lot clearer on PIL-A5. It could be because it's less flexible and therefore we have improved resolution here. So we went on to identify the sugars, and first of all we did a lectin binding experiment, which works as follows. So first, you can separate your purified pili on an STS page gel. The position of PIL-A4 is indicated with blue arrowhead, PIL-A5 with an orange arrowhead. You then incubate the gel with biotinylated lectins. Lectins are sugar binding proteins, so they will bind to specific sugars to which they target. The gel is then incubated with streptavidin fluorescent dye, and we had a positive result for these 3 lectins, which shows us that we have the presence of mannose and or glucose and N-acetyl galactosamine. So these are likely the sugars that account for that density in our EM maps. However, we wanted a bit more detail on this, so we engaged with the company Ludger in Oxford, and they removed the sugars from our pili and performed ultra high performance liquid chromatography and tandem mass spectrometry. And these are the resulting traces. And we don't need to, again, worry about too much of the detail. The important point is that both the wild type sample with both filaments and the mutant with only wide filaments, the traces were very similar. So it looks like that both filaments are probably modified in the same way. And there were 2 peaks of 2 species, which are by far more abundant than any others. And the structures of those are shown here. So the PROC part we can ignore. This is a fluorescent part which is used for detection. But you can see here, these are likely to be the structures of the 2 sugars that constitute this glycosylation. The hexose is likely to be glucose or mannose because that was confirmed in our lectin binding experiments, and the N-acetyl hexosamine is likely to be N-acetyl galactosamine. This was again confirmed by our lectin binding experiments. Of interest, we also identified an unknown carbohydrate which has not been seen before in bacteria, and we still don't know what the structure of this is. So the role of glycosylation may have many functions. So it could prevent pilus bundling, enhance the stability of filaments, increase adhesive properties, which is very important both in biofilm formation, motility, also for cell-cell and cell environment communication. And in some species, they've been shown to act as a defense mechanism for phage infection. Okay, so I'm going to end on a bit of speculation. And this requires us to have a look at the charge patterns in both filaments. So PIL-A4 has a positively charged patch winding around the filament axis, which is not found in PIL-A5. To remind you, PIL-A4 is the filament which we think is involved in DNA binding and uptake. And we can dock a double-stranded DNA molecule into this patch of residues, and it conveniently avoids the glycosylation. But as I said, it's pure speculation, and it now remains to be tested. We also, as I mentioned in the introduction, don't really know how this process works. And whilst other groups have also suggested that DNA could bind to the filaments, recently some other groups have shown that DNA may bind to the tip of filaments. But what we can say in our hands that certainly when we mutate PIL-A4, we don't have any DNA uptake anymore. So it seems like the wide filaments are required for natural transformation. But conversely, there are some mutants which are impaired in piliation but are hypertransformable. So there's still a lot more to be done to understand this mechanism. Our current working hypothesis is that the wide pili may capture DNA rather like a fishing net, which would increase the local concentration of DNA near the outer membrane and thus improve the efficiency of DNA uptake. So to summarize, I've shown you that Thermus can assemble both a wide and a narrow type 4 Pilus filament, and that they're composed of different proteins and have differences in their structural assembly, surface charge, hydrophobicity, and function. We find that PIL-A4 is required for DNA uptake, whereas PIL-A5 is required for motility. We find both filaments are glycosylated with long sugar chains. It'll now be interesting to investigate if this is a feature in other bacteria. So can cells assemble different types of filament to perform unique functions? And if so, it will be then interesting to go on to investigate if we can, for example, develop drugs to target specific mechanisms, particularly important for pathogenic bacteria. So I would like to acknowledge a few people. So as I said, I started the work in the Kulbrandt lab in Frankfurt, where I'd like to acknowledge Selva, who started the work on the helical reconstruction. All of this work was done in collaboration with the Averhof lab at the University of Frankfurt. That was initially Ralph, then Kirsten, and then more recently Lennart. In Exeter, I want to acknowledge my postdoc Alex Neuhaus, who did the bulk of the work on helical reconstruction, and also Bertram, Kelly, and Matt for all of their contributions. Lastly, there's my group. I'd like to acknowledge them for their hard work and collaboration. The work was funded by the BBSRC. Again, I'd also like to acknowledge Diamond for the Cryos time for collection of this data set. And hopefully this work will be published perhaps this week if we're lucky. So that's quite nice timing. And yeah, I'm happy to take any questions.
Well, thanks a lot, Vicky, for a really great talk. So there are quite a few questions for you. We don't have a deal of time, so I think one of the questions was what software did you do the processing in?
Shall I do just a minute? I just stopped my screen. Okay.
So one of the questions was what did you do the processing in, and how did you distinguish between open and closed states by the 2D classification?
So for the tomography, the open and closed state, this was done with iMod and Pete. And distinguishing between the two, well, they were manually picked, so we could see which ones had pili and which ones didn't, and separate them that way.
Cool, thank you. So one question was, why did you focus on the thermal bacteria instead of the Pseudomonas pili you showed in your introduction? What was the benefit of this sample for cryo-ET?
Well, historically, we were already working with Thermus, so practically it was something that we just started investigating, and then we found something interesting. We found these two different Pilus types, so it just sprung up from there.
Let me see. I think that, yeah, I think there's just too many. Maybe we'll try and cover some. Well, maybe we can't, but we'll see. Let me think. There was a question about pipetting the bacteria onto the grids. Did you have to wait for them to reattach before freezing?
So actually, in some cases, we developed well, not really developed. We just did a slightly different way of preparing the grids, because if you pipette cells with pili, sometimes they can shear off. So we sometimes actually just dipped grids into solutions of bacterial cells, and they then stuck to the surface. We didn't have to wait any kind of length of time. We then just plunge froze them.
Well, thanks a lot for answering the questions and for a really great talk. As I say, there are a lot more, but unfortunately, we're going to have to kind of move on to the next speaker. Yeah, so thanks, Vicki.
You're welcome.